Picture a dragonfly with wings wider than your outstretched arms, wider than the handlebars of a bicycle, wider than some hawks and owls that rule the skies today. Imagine a millipede as long as a car, longer than most adult humans are tall, crawling through forests of towering ferns and scale-barked trees under an alien sky. Envision a scorpion the size of a house cat, stalking through ancient swamps under an atmosphere so rich in oxygen that the very air would feel intoxicating to our modern lungs, heavy and almost electric with life-giving gas. Picture these creatures not as fantasy, not as monsters from mythology or creatures of fevered imagination, but as real animals that breathed, that hunted, that mated and reproduced, that struggled and thrived for millions upon millions of years in a world that was fundamentally different from anything we know today. These creatures were not myths plucked from ancient legends or monsters conjured by creative minds seeking to terrify. They were real. They walked and crawled and flew across real landscapes. They left real footprints in real mud that hardened into real stone. Their bodies, or parts of them, were preserved in sediments that compressed over unfathomable stretches of time into the fossils that paleontologists now study with wonder and careful scientific rigor. They breathed the same elements we breathe: carbon and oxygen, nitrogen and trace gases, though in dramatically different proportions. They hunted prey, evaded predators, competed for mates and territory, experienced hunger and satisfaction, exhaustion and rest, all the fundamental experiences that define animal life. And for millions of years, longer than humans have existed as a species, longer than most mammal lineages have walked the earth, these giants ruled their world with a dominance that seems almost impossible when we look at the small, delicate insects that share our modern landscapes. Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. If you like these videos, please consider subscribing to the channel and commenting below if you have any feedback or suggestions for topics of future episodes. Tonight, we journey back over 300 million years to a time when insects reached sizes that would dwarf any creature with 6 legs alive today, when arthropods in general pushed the boundaries of their body plans to limits that seemed to defy the laws of physics and biology as we understand them from modern observations. To understand why these giants existed and why they vanished so completely that only their fossils remain to tell their story, we need to step into their world completely, to immerse ourselves in the conditions that made their existence possible. We need to feel the weight of that ancient atmosphere pressing down with slightly greater force than our modern air, to understand the chemistry that flowed through their tracheal tubes and powered their enormous bodies. We need to see through the compound eyes of predators that commanded the skies long before birds existed, before pterosaurs glided on leathery wings, before any vertebrate had mastered the art of powered flight. We need to witness an Earth in the midst of transformation, where the very air we breathe held different chemistry, different possibilities, and different dangers, where the rules of life were written differently than they are today. The story of giant insects is not just about size, not merely a curiosity of scale, a footnote in the history of life, noting that insects were once larger and are now smaller. It is about what happens when a planet's fundamental conditions shift, when the invisible composition of the atmosphere that mixture of gases we take for granted with every breath, can determine whether a creature thrives or perishes, whether a body plan succeeds spectacularly or fails completely. It is about ecological niches standing empty, waiting with infinite patience for evolution to fill them in ways that seem impossible by today's standards, that violate our instincts about how large insects can grow. These were the days when arthropods pushed the boundaries of what their segmented bodies, their jointed legs, their exoskeletons of chitin and protein could achieve, creating forms so extraordinary that when their fossils were first discovered in coal mines and quarries across Europe and North America, scientists struggled to believe what they were seeing, struggled to accept that insects, those small creatures we swat away without thought, could once have been large enough to prey on vertebrates, large enough that their footsteps left trackways visible in stone. But the evidence is undeniable, preserved in multiple independent lines of investigation that all point to the same conclusion. It is in stone, in the fine impressions of wing veins preserved in shale, in the trackways left in ancient mud, in the fragmentary remains of exoskeletons that once covered bodies larger than we can easily imagine. It is in the delicate impressions of wings that once carried predators through humid forests beneath a richer sky, wings reinforced with intricate patterns of veins that an engineer today would recognize as elegantly designed structures, balancing strength with lightness, rigidity with flexibility. And it is in the chemistry of ancient atmospheres, reconstructed painstakingly from the composition of air bubbles trapped in amber, from the ratios of isotopes in ancient soils, from computer models that simulate the complex feedback loops between plants, decomposers, ocean chemistry, and atmospheric composition over geological time. Let us begin where all stories of ancient giants must begin, with the air itself, with the invisible substance that surrounds us, that we breathe without thinking, and with the element that made the impossible real, that allowed insects to grow to sizes that today would be physiologically unfeasible. Oxygen, the molecule of life, and the key to understanding why 300 million years ago, the world belonged to creatures that seem to us like monsters from a dream. The late Carboniferous and Early Permian periods, spanning roughly from 359 million to 252 million years ago, witnessed Earth under conditions radically different from our modern world. The atmosphere itself was transformed, not just in subtle ways, but fundamentally at a chemical level that would reshape what life could become. Today, we live in air that contains approximately 21% oxygen. This concentration feels normal to us because we evolved within it, our lungs and metabolic systems calibrated over millions of years to extract precisely what we need from this mixture of gases. Our respiratory systems, our cardiovascular networks, even the size and structure of our cells are all tuned to function optimally at this specific oxygen level. It is, for us, the baseline, the standard against which all other conditions seem abnormal. But travel back to those ancient times, and you would find yourself breathing air far richer in oxygen, with concentrations reaching 30 to 35%, an increase of nearly 50% more than what sustains us now. To a human transported back to that era, the air would feel thicker, almost intoxicating. Your lungs would work with unusual ease, each breath delivering more oxygen than you are accustomed to. You might feel energized at first, then overwhelmed, as the excess oxygen began to stress your system in ways it was never designed to handle. For us, that atmosphere would be toxic in extended exposure. But for the creatures that evolved within it, it was paradise. This difference may seem abstract until you understand what it meant for the creatures living then. Oxygen is the fuel that powers movement, growth, and metabolism. It is the molecule that allows cells to extract energy from food, converting sugars and fats into the chemical energy that drives muscle contractions, nerve signals, and all the myriad processes that keep an organism alive. The more readily available oxygen is, the more energy organisms can generate, the faster their metabolisms can run, and the larger their bodies can grow without running into the limits imposed by respiratory efficiency. And for insects, this abundance of oxygen unlocked possibilities that the laws of physics and biology would otherwise forbid. To understand why oxygen mattered so profoundly for insects, far more than for vertebrates, we need to look at how insects breathe, because their respiratory system is fundamentally different from ours, and it is this difference that both enabled their ancient gigantism and limits their size today. We mammals breathe through lungs, organs specifically evolved to maximize surface area for gas exchange. Air enters our lungs, fills millions of tiny sacs called alveoli, and oxygen diffuses across thin membranes into our bloodstream. From there, red blood cells, packed with hemoglobin molecules, bind to the oxygen and transport it throughout the body via a complex circulatory system powered by the heart. This is an active, pumped system capable of delivering oxygen to tissues far from the lungs, even to the extremities of large animals. It is efficient, adaptable, and scalable, which is why mammals and other vertebrates can grow to enormous sizes. A whale, weighing many tons, can still deliver adequate oxygen to every cell in its body, because its respiratory and circulatory systems are designed for that task. Insects do not have lungs. They do not have blood that carries oxygen to their tissues in the way ours does. They have hemolymph, a fluid that circulates through their bodies, but it does not play a primary role in oxygen transport. Instead, insects breathe through a network of tiny tubes called tracheae, which branch throughout their bodies like an intricate system of air pipes, reaching into every tissue, every organ, every cell that requires oxygen. These tracheal tubes begin at small openings along the sides of the insect's body called spiracles. If you look closely at the abdomen of a large insect, like a grasshopper or a beetle, you can sometimes see these spiracles, tiny pores that open and close, regulating the flow of air in and out of the body. Oxygen enters through these spiracles and travels down the tracheae, which branch repeatedly, becoming finer and finer, until they terminate in tiny tubules called tracheoles that make direct contact with individual cells. The oxygen diffuses directly from the air in these tubes into the cells, and carbon dioxide, the waste product of metabolism, diffuses back out. This is passive diffusion, driven entirely by the concentration gradient between the oxygen-rich air outside and the oxygen-poor tissues inside. There is no pumping in the traditional sense, no heart-forcing oxygenated fluid through vessels. Some insects can assist the process by contracting their abdominal muscles to compress and expand their tracheal system, creating a gentle ventilation. But the fundamental mechanism is diffusion, the spontaneous movement of molecules from areas of high concentration to areas of low concentration. This system works beautifully for small insects. The distances involved are short, often just millimeters, and oxygen can diffuse through the tracheal tubes and reach every cell efficiently, A fruit fly, measuring just a few millimeters in length, has no difficulty obtaining all the oxygen it needs. Its tracheal system is simple, the pathways short, and the surface area to volume ratio is favorable. But as an insect grows larger, the physics become problematic, and this is where the fundamental constraint appears. The volume of tissue requiring oxygen increases much faster than the surface area available for gas exchange. This is a consequence of geometry, an inescapable mathematical relationship that applies to all three-dimensional objects. If you double the length of an insect, you increase its volume eightfold, because volume scales with the cube of linear dimensions. But you only increase its surface area fourfold, because area scales with the square of linear dimensions. The inside of a large insect, the core of its body, is much farther from the outside air than the inside of a small insect, and simple diffusion becomes inadequate. Oxygen must travel greater distances through the tracheal tubes to reach the innermost tissues, and the concentration gradient weakens the farther you go from the spiracles. The core of the insect's body would begin to suffocate, unable to receive enough oxygen to power its muscles and organs. This is why today's insects are relatively small. The modern atmosphere, with its 21% oxygen, imposes a strict size limit. Push beyond that limit, and the insect cannot sustain itself. Its metabolism would slow, its muscles would weaken, and it would die. But in the late Carboniferous, with oxygen concentrations reaching 35%, that limit was pushed dramatically higher. The steeper concentration gradient meant oxygen could diffuse farther and faster through those tracheal tubes. The difference in oxygen concentration between the air at the spiracles and the oxygen-depleted tissues deep within the body was much greater, driving diffusion more efficiently. An insect could grow larger, its tissues farther from the surface, and still receive adequate oxygen to maintain an active lifestyle. The very air itself became a resource that could support bodies of astonishing proportions. This was not a small adjustment, a minor tweak to body size. This was a transformation of what was physically possible, a lifting of constraints that had kept insects small for tens of millions of years. And there was another factor at play, one that mattered especially for flying insects. Higher oxygen levels also meant a denser atmosphere overall, because atmospheric pressure increases with the concentration of gases. This denser air provided more lift for wings, making it easier for larger, heavier insects to achieve and sustain flight. Aerodynamic forces scale with air density, so in that thicker atmosphere, even massive insects could take to the skies with less energy expenditure than would be required today. Flight, one of the most metabolically demanding activities any organism can undertake, became more feasible for creatures that would be grounded under modern conditions. The Carboniferous forests themselves contributed to this oxygen-rich world. This was the age of vast coal swamps, where towering club mosses and giant horsetails created dense, humid forests unlike anything we see today. These ancient plants photosynthesized at tremendous scales, releasing oxygen into the atmosphere as a byproduct. But crucially, the mechanisms for decomposition were not yet fully developed. Fungi and bacteria that could efficiently break down lignin, the tough substance that gives wood its strength, had not yet evolved. When these giant plants died, they did not decay rapidly. Instead, they accumulated layer upon layer in the swampy ground, locking away the carbon they had captured from the atmosphere and leaving the oxygen behind. Over millions of years, this imbalance drove oxygen concentrations upward, creating the hyperoxic conditions that would define the era. It was in this world, under these skies, that insects began to grow. And none grew more spectacularly or more fearsome than the griffinflies, the ancient relatives of modern dragonflies that would come to dominate the airways of the Paleozoic. The first fossil of Meganeura was discovered in 1880 in the coal mines of Commentry, France. Workers extracting coal from deep underground, in beds that had once been ancient swamps, began finding impressions preserved in the rock, pressed between layers of compressed plant material that would eventually become the coal that fueled the Industrial Revolution. These were not ordinary fossils. They were the delicate, intricate imprints of insect wings, but wings of a size that seemed impossible. Wings that suggested creatures far larger than any insect alive in the modern world. The finest specimen revealed a creature with a single wing length reaching 32 centimeters, 13 inches, and a total wingspan estimated between 65 and 75 centimeters, roughly 2.5 feet across. To put this in perspective, that is wider than the wingspan of many hawks and owls, wider than most of the smaller eagles, wider than any flying insect that exists today by a factor of 3 or 4. This was Meganeura monii, and when French paleontologist Charles Brongniart described it in 1885, naming it for the large veins that networked across its wings like the veins of a leaf, intricate patterns that had been preserved with extraordinary fidelity in the fine-grained shale. It became the iconic image of prehistoric insect gigantism. The name Meganeura translates to large vein, a simple, descriptive term that captures one of the most striking features of these creatures. The wing venation, the pattern of support structures that gave the wings their strength and flexibility, was far more complex than that of modern dragonflies, with dense cross-bracing and reinforcing veins that allowed these massive wings to withstand the stresses of powered flight. These were not fragile structures. They were engineering marvels, thin membranes stretched over a framework that could support the forces generated by rapid wing beats and aerial maneuvers. Imagine encountering such a creature in flight, not as a fossil, not as an illustration, but as a living animal in its native habitat. Picture yourself standing at the edge of one of those ancient swamps, the air thick and humid, moisture condensing on every surface, the temperature warm and constant, the canopy above alive with the rustling of giant ferns and the hum of countless insects going about their lives, feeding, mating, avoiding predators. The light filters down through the dense vegetation, green and diffuse, and the ground beneath your feet is soft, waterlogged, covered in a mat of decaying plant material that releases the earthy smell of decomposition. You hear the calls of amphibians, strange croaking and bellowing sounds from creatures that are neither quite fish nor quite fully adapted to land. And you hear the rustle of movement in the undergrowth, arthropods of various sizes going about their business. Then, cutting through that soundscape, comes something different. A low thrumming beat, rhythmic and powerful, growing louder. You look up, and into view comes a predator unlike any dragonfly you have seen in the modern world. Its body is long and slender, measuring perhaps 40 centimeters, nearly 1.5 feet from head to tail, segmented and armored, built for durability and power rather than delicate grace. Its wings, 4 of them arranged in 2 pairs, stretch out in a blur of motion, each membrane reinforced by that dense network of veins you saw in the fossils. And they beat with a force you can feel in the air, creating downwash and turbulence that stirs the vegetation below. And its eyes, those enormous compound eyes, dominate its head, bulging outward to provide an almost spherical field of vision. Thousands of individual lenses working together to create a mosaic image of the world. These eyes could see in nearly every direction at once, tracking movement above, below, to the sides, behind, providing the kind of situational awareness that modern fighter pilots spend millions of dollars trying to achieve with technology. For a flying predator, this was an extraordinary advantage, allowing it to spot prey from great distances and coordinate its movements with devastating precision, to judge distances accurately, to time its strikes perfectly, to avoid obstacles even at high speed. The legs of Meganeura were equally specialized, adapted not for walking, though they could support the insect when it rested, but for catching. Along the tibiae and tarsi, the lower segments of its legs were rows of sharp spines, arranged in patterns that suggest careful evolutionary refinement. These were not random projections. They were precisely placed structures, designed to interlock, to grip, to hold fast. When Meganeura hunted, it would patrol through the open spaces above rivers and wetlands, using its large eyes to scan the environment below. It flew high, where visibility was good, where it could see across vast stretches of habitat, and it watched for movement, a flash of wings, the glint of sunlight off a wet body, the erratic flight of a smaller insect trying to escape some other threat. Any of these would trigger the hunt. Upon spotting prey, perhaps a large paleodictyopteran with its own impressive wingspan, or a mayfly, or even a smaller predatory insect, Meganeura would accelerate with powerful wing beats, its body angling downward, its trajectory calculated with the precision that comes from millions of years of evolutionary refinement. As it closed the distance, traveling at speeds that may have exceeded 30 kilometers per hour, 20 miles per hour, according to analyses of its wing structure and body mass, it would reach out with those spiny legs, extending them forward and spreading them wide. The moment of contact was decisive. Those legs would snap together like a cage, the spines interlocking, gripping the prey's body or wings, and the struggling victim would find itself held fast, unable to escape, unable to fight back effectively. Then came the final act. The powerful mandibles at Meganeura's mouth, toothed jaws built for tearing and crushing, would clamp down on the prey's head or thorax, often killing it instantly or rendering it incapable of resistance. And Meganeura would carry its meal to a perch, perhaps a branch overhanging the water, perhaps a log on the ground, and there it would feed, methodically dismantling its prey, extracting the nutritious soft tissues and discarding the harder parts. Fossil evidence suggests that these griffinflies were apex predators of the insect world, occupying a niche similar to that of hawks and falcons in modern ecosystems. They were generalist hunters, capable of taking a wide variety of prey, and their size allowed them to target animals that smaller predators could not handle. Some paleontologists have even suggested that Meganeura occasionally preyed on small vertebrates, specifically the early amphibians that were just beginning to colonize the land and were still relatively vulnerable, particularly in their juvenile stages. A young amphibian, freshly metamorphosed and venturing onto land for the first time, would have been an easy target for a predator with the speed and striking power of Meganeura. There are no fossilized gut contents to confirm this directly, as preservation of such details is extremely rare. But the anatomy of Meganeura, particularly the strength of its jaws and the robustness of its legs, is consistent with an animal capable of handling prey larger and more robust than typical insect fare. But Meganeura, as impressive as it was, would eventually be surpassed. In 1939, paleontologist Frank Carpenter described a new species from fossils found in Kansas, in the early Permian rocks of the Wellington Formation. This was Meganeuropsis permiana, and it would hold the record as the largest flying insect ever known. Carpenter's initial description was based on an incomplete wing, but the size of that single fragment was staggering. When reconstructed, Meganeuropsis was estimated to have a wingspan reaching 71 centimeters, 28 inches, and a body length of up to 43 centimeters, 17 inches. Some estimates push the wingspan even higher, to 75 centimeters, roughly the span of a crow or a small hawk. This creature, by every measure, was a giant. What allowed Meganeuropsis to reach such extraordinary dimensions? The answer lies partly in timing. Although Meganeuropsis lived during the early Permian, a time when oxygen levels had begun to decline from their Carboniferous peak, the atmospheric concentration was still significantly higher than today, estimated at 23 to 30%. This was enough to sustain large body sizes, especially for an insect whose body was not particularly heavy. Despite their massive wingspans, griffinflies like Meganeuropsis had lightweight, slender bodies. Their exoskeletons were thin, their internal structures delicate, and their wings, though large, were membranes stretched over a framework of veins. They were giants of wingspan, but not of mass. Studies comparing them to modern beetles suggest that even the largest griffinflies weighed less than some of today's heaviest insects, such as the Goliath beetle, which can reach over 100 grams. Estimates for Meganeuropsis place its weight somewhere between 100 and 150 grams, roughly a third of a pound, light enough that flight remained energetically feasible. The structure of their wings was key to their success. Unlike the wings of true dragonflies, which would evolve later, griffinfly wings lacked certain specialized features. They had no pterostigmata, the thickened spots near the wingtips that help stabilize dragonfly flight. Their wing venation was simpler, with fewer cross braces. This suggests that their flight, while powerful, was less maneuverable than that of modern dragonflies. They were built for straight-line speed and endurance, not for tight turns and aerial acrobatics. This made them ideal hunters in open environments, where they could patrol long stretches of rivers and lake shores, covering great distances in search of prey, but less suited to dense forests where quick changes in direction would be necessary. Fossil evidence supports this ecological interpretation. The best preserved specimens of griffinflies come from sediments that represent open habitats, near water bodies, with scattered vegetation, rather than dense canopies. These were the hunting grounds where size was an advantage, where a large predator could outpace smaller competitors and prey, and where aerial agility was less critical than raw power and visual range. The compound eyes of these creatures were enormous, with thousands of individual lenses, giving them exceptional visual acuity. They could spot a moving insect from meters away, lock onto it, and close the distance before their prey could react. In an ecosystem where they were among the largest flying creatures, they had little to fear from above. The skies belonged to them. Their nymphs, the juvenile forms, lived in water, just as modern dragonfly nymphs do. Fossils of griffinfly nymphs reveal that they too were predators, with strong, grasping mouthparts and powerful bodies adapted for ambush hunting in ponds and slow-moving rivers. They likely fed on smaller aquatic insects, amphibian larvae, and even small fish. The transition from water to air through metamorphosis was a complete transformation, but the predatory lifestyle remained constant. From the murky depths to the open sky, these were creatures built to hunt. But griffinflies were not the only giants walking and crawling through the Carboniferous forests. In fact, the largest land arthropod of all time was not an insect at all, but a distant relative of modern millipedes, and its fossils tell a story of life pushed to the absolute limits of what a segmented, many-legged body could achieve. Arthropleura is a name that carries weight, quite literally, and not just because of the immense size of the creature it describes, but because of what that size represents, the absolute limits of what a terrestrial arthropod body plan could achieve under the conditions of the Carboniferous and Early Permian. This genus of extinct millipede-like creatures reached lengths that still astonish scientists today, with the largest individuals estimated at 2.6 to 2.7 meters long, nearly 9 feet, and weighing approximately 50 kilograms, over 100 pounds. To put this in perspective, Arthropleura was as long as a small car, longer than most adult humans are tall, and as heavy as a large dog. When you imagine it moving through the undergrowth of a Carboniferous forest, you are picturing an animal that dwarfed nearly every other creature on land at the time. An animal whose footsteps would have been audible, whose presence would have been unmistakable. Fossils of Arthropleura have been known to science since 1849, when the first fragments were discovered during railway construction near Friedrichsthal in Germany. Unearthed as workers cut through layers of ancient rock to lay tracks for the expanding rail network of the Industrial Revolution. Over the decades, additional specimens have been found across Europe and North America, in coal mines and quarries, in riverbeds and cliff sedges, anywhere that Carboniferous and Early Permian rocks are exposed to the surface. But despite over a century and a half of searching, complete fossils are extraordinarily rare, so rare that for most of the 20th century, paleontologists had only fragmentary remains to work with. Isolated plates of exoskeleton, pieces of legs, sections of body segments, enough to know that something enormous once lived, but not enough to fully reconstruct its appearance or understand its biology in detail. The reason for this rarity lies in the nature of Arthropleura itself, in the fundamental design of its body. Like modern millipedes and centipedes, its body was composed of many segments, perhaps 30 in total, each covered with plates of exoskeleton made of chitin and proteins, hard but not mineralized, strong enough to protect the soft tissues beneath, but lightweight enough not to burden the animal with excessive mass. These segments were connected by softer, flexible tissue, allowing Arthropleura to move with a wave-like motion, its body undulating as it walked. But when an Arthropleura died, this very design became a problem for fossilization. The body would quickly disarticulate, the segments separating and scattering as decomposition progressed, and scavengers fed on the remains. The soft connective tissues would decay first, and then the individual plates of exoskeleton would disperse, carried by water currents if the animal died near a river, or simply crumbling in place if it died on dry land. The result is that most Arthropleura fossils consist of isolated plates, fragmentary sections, or, most commonly and most tantalizingly, trackways. These trackways are remarkable in their own right, windows into behavior that body fossils alone cannot provide. In places like Scotland and Nova Scotia, paleontologists have discovered fossilized trails left by Arthropleura as it moved across ancient mud and sand, environments that were soft enough to record the impressions of its feet, but stable enough and buried quickly enough by subsequent sedimentation to preserve those impressions for hundreds of millions of years. These tracks can be up to 50 centimeters wide, 20 inches, broader than the span of a human hand, and they reveal important details about how the animal moved, details that complement what we can infer from the anatomy of the body fossils. The pattern of impressions shows that Arthropleura had many legs, perhaps 32 to 64 in total, arranged in pairs along each body segment, and that these legs were short and closely spaced. The stride length, the distance between successive footprints, is relatively short, suggesting that Arthropleura was a slow mover, likely incapable of the rapid escape bursts that some modern arthropods can achieve. It was not built for speed. It was built for steady, methodical progress, for covering ground patiently, for endurance rather than agility. The trackways also show that Arthropleura traveled alone rather than in groups. While some modern millipedes aggregate in large numbers, sometimes thousands of individuals moving together, Arthropleura tracks are typically solitary, single trails crossing the ancient landscape with no companions. This suggests a relatively solitary lifestyle, perhaps with individuals maintaining territories, or simply spreading out across the available habitat to avoid competition for food. And those trackways sometimes show signs of resting behavior, places where the trail widens and deepens, where the animal may have stopped, curled up, and remained stationary for a period, before resuming its journey. These resting sites are poignant, small windows into the daily life of a creature that lived so long ago, evidence that even giants need to rest. The most significant recent discovery came in 2018, when a fossil was found at Howick Bay, near Newcastle in England, on a beach where ancient cliffs are constantly eroding, exposing new layers of rock and, occasionally, revealing treasures that have been hidden for hundreds of millions of years. A geologist on a casual walk, someone who happened to know what to look for and happened to be in the right place at the right time, noticed a boulder that had fallen from the cliff face, and within it, embedded in the sandstone, was a section of Arthropleura exoskeleton, approximately 75 centimeters long, nearly 2.5 feet. This was not a complete specimen, but it was large, far larger than most Arthropleura fossils, and remarkably well preserved, with details of the surface texture, the articulation of the plates, the arrangement of the segments, all clearly visible. Based on the proportions of this fragment and comparisons with other, more complete but smaller specimens, researchers estimated that the complete animal would have measured 2.7 meters in length, making this the largest Arthropleura individual known, and by extension, the largest land arthropod ever discovered. This fossil provided crucial new information about Arthropleura's anatomy and lifestyle. The preservation quality was exceptional, allowing researchers to examine fine details of the exoskeleton that had never been visible before. They could see the texture of the surface, a pattern of fine ridges and grooves that may have served to strengthen the plates or provide attachment points for muscles. They could see how the segments fit together, overlapping slightly to allow flexibility while maintaining protection. And they could infer from the geological context where and how this animal lived. The Howick Bay specimen was found in sediments that represent an ancient river channel, layers of sandstone and silt deposited by flowing water over 300 million years ago. This was not a coal swamp, not the dense, waterlogged forest environment that had been assumed to be Arthropleura's primary habitat. This was an open woodland, with rivers and streams, with vegetation, but not the overwhelming density of the coal forests, a drier, more varied landscape. This discovery has reshaped our understanding of where Arthropleura lived. It was not confined to swamps. It inhabited a range of environments, anywhere there was abundant plant litter and moisture, from the wettest forests to relatively open woodlands near rivers. A creature of Arthropleura's size would have had difficulty navigating through the densest vegetation, where low-hanging fronds and tangled undergrowth would impede movement. Open woodlands with scattered trees and plenty of ground-level plant litter would have offered abundant food and room to move, space for a giant to exist without constantly fighting through obstacles. One of the long-standing questions about Arthropleura has been its diet. Early interpretations, based on the assumption that such a large arthropod must have been a predator, suggested that it had strong carnivorous mouthparts. However, no Arthropleura fossil has ever been found with a preserved head, which has made direct examination of its feeding structures impossible. Instead, scientists have relied on indirect evidence. Fossilized gut contents and coprolites, fossilized feces associated with Arthropleura, have been found to contain plant material, including fragments of ferns and spores from lycophytes. This strongly suggests that Arthropleura was a detritivore, feeding on decaying plant matter, much like modern millipedes. It likely roamed the forest floor, processing fallen leaves, rotting wood, and other organic debris, playing a key role in nutrient cycling within those ancient ecosystems. The discovery of the Howick Bay specimen also provided insights into Arthropleura's habitat preferences. The fossil was found in sediments that represent an ancient river channel, suggesting that Arthropleura lived in open woodlands near water, rather than in the dense coal swamps that were once thought to be its primary habitat. This makes ecological sense. A creature of Arthropleura's size would have had difficulty navigating through dense vegetation. Open woodlands with scattered trees and plenty of ground-level plant litter would have offered abundant food and room to move. The exoskeleton of Arthropleura was segmented and jointed, allowing for flexibility despite the animal's size. Each segment was covered by 3 plates, one on top and one on each side, forming a protective shell. The dorsal plates, those on the back, could extend outward slightly, creating a flared appearance that may have made Arthropleura look even larger and more intimidating. The coloration of these plates is, of course, unknown, but some reconstructions suggest patterns that would have provided camouflage among the leaf litter, helping Arthropleura blend into its surroundings despite its size. One of the most intriguing aspects of Arthropleura is its evolutionary position. For many years, it was classified simply as a giant millipede, but recent studies of juvenile fossils have revealed surprising details. In 2024, researchers announced the discovery of two small, well-preserved Arthropleura fossils from France, measuring just 25 and 40 millimeters in length. These juveniles provided the first clear view of Arthropleura's head, and what they found was unexpected. The head had features of both millipedes and centipedes. It had enclosed mandibles and two pairs of head appendages positioned behind them, characteristics more typical of centipedes. This discovery suggests that millipedes and centipedes may be more closely related than previously thought, and that Arthropleura represents an early branch in their evolutionary tree, a time before these groups fully diverged. But perhaps the most evocative evidence of Arthropleura's existence comes not from body fossils, but from burrows. Large, tunnel-like structures have been found in Carboniferous sediments, and some researchers believe these may have been created by Arthropleura during periods of estivation, a form of dormancy similar to hibernation. These burrows would have provided shelter during dry seasons or unfavorable conditions, allowing Arthropleura to survive times when food or water was scarce. The image of a giant millipede curled up in an underground chamber, waiting out the climate, is both strange and oddly familiar, a behavior echoed by many modern invertebrates. Arthropleura thrived for tens of millions of years, from the early Carboniferous to the early Permian, but eventually it vanished from the fossil record. The reasons for its extinction are still debated, but likely involve a combination of environmental changes and competition. As oxygen levels began to decline, maintaining such a large body would have become increasingly difficult. The Carboniferous rainforest collapse, a period of climate change that transformed vast swamp forests into drier, more seasonal environments, would have reduced the availability of the moist, plant-rich habitats Arthropleura depended on. Additionally, the rise of early reptiles, which were more mobile and adaptable than the slow-moving Arthropleura, may have introduced new predators and competitors that the giant millipede could not withstand. By the end of the early Permian, Arthropleura was gone, leaving behind only its fossils and trackways as evidence of a time when millipedes could grow to the size of automobiles. Yet, Arthropleura was not the only arthropod pushing the limits of size in those ancient times. The land and waters of the Carboniferous and Permian were filled with creatures that would seem monstrous by modern standards, including scorpions that rivaled house cats and aquatic predators that dwarfed any living arthropod. In the swamps of what is now Scotland, 335 million years ago, lived a scorpion that still captures the imagination of paleontologists. This was Pulmonoscorpius kirktonensis, discovered in 1994 at the East Kirkton Quarry in West Lothian. Pulmonoscorpius, whose name means breathing scorpion, was enormous by scorpion standards, reaching lengths of up to 70 centimeters, 28 inches. That is longer than most domestic cats, and this was a venomous predator with powerful pincers and a segmented tail ending in a stinger. Fossils of Pulmonoscorpius are exceptionally well preserved, with over 16 complete specimens and more than 300 additional fragments recovered from East Kirkton. These fossils were preserved in fine limestone and shale, deposited in ancient lake beds, and they reveal extraordinary details about the scorpion's anatomy. The fossils consist only of the outer layer of the exoskeleton, a thin, transparent cuticle, estimated to be just 15 to 18 micrometers thick. Despite this delicate preservation, the fossils capture fine structures, including the surface texture of the carapace, the arrangement of sensory hairs, and the intricate details of the legs and pincers. Pulmonoscorpius had a body plan similar to modern scorpions, but with some notable differences. Its lateral eyes were large, far larger than those of most modern scorpions, which tend to be nocturnal and rely less on vision. The size and structure of these eyes suggest that Pulmonoscorpius was a diurnal hunter, active during the day, using its keen eyesight to locate prey. Its prosoma, the front section of the body, was covered by a carapace bearing both median eyes near the center and compound lateral eyes on the sides. Each lateral eye contained 40 to 60 individual lenses, providing wide-angle vision and excellent motion detection. The pedipalps, the large pincer-like appendages at the front of a scorpion, were robust in Pulmonoscorpius, but not overwhelmingly so. In modern scorpions, there is a trade-off between pincer strength and venom potency. Species with large, powerful pincers tend to have milder venom, relying on mechanical force to subdue prey. Species with smaller pincers often have more potent venom, using chemical means to immobilize their victims. Pulmonoscorpius had moderately sized pincers and a proportionally large tail, suggesting that it relied heavily on its venom. The telson, the bulb at the end of the tail containing the venom gland, was well developed, with strong carinae or ridges on its underside, likely for added strength when striking. The diet of Pulmonoscorpius is not known from direct evidence, but based on its size and anatomy, it likely preyed on smaller arthropods and possibly small tetrapods, the early amphibians and reptiles that were just beginning to colonize the land. The spines on its legs would have helped it grip struggling prey, and its venom would have subdued victims quickly. Pulmonoscorpius lived in a time when competition for food was intense, and being a large, versatile predator would have been a significant advantage. Interestingly, Pulmonoscorpius possessed 4 pairs of book lungs, respiratory structures that allow scorpions to breathe air. These book lungs are stacks of thin, leaf-like membranes filled with blood that absorb oxygen from the air and release carbon dioxide. The presence of book lungs in Pulmonoscorpius confirms that it was fully terrestrial, unlike some of its earlier relatives, which were aquatic or amphibious. This adaptation to land, combined with the high oxygen levels of the Carboniferous, allowed Pulmonoscorpius to reach sizes that would be impossible for scorpions today. Modern scorpions rarely exceed 20 centimeters in length, less than a third the size of their ancient ancestor. But, as impressive as Pulmonoscorpius was on land, the largest arthropods of the Paleozoic lived in the water. These were the Eurypterids, often called sea scorpions, though many lived in fresh or brackish water rather than the sea. And among the Eurypterids, one genus stands above all others in size. Jacalopterus rhenanii, discovered in early Devonian rocks of Germany, is estimated to have reached lengths of 2.3 to 2.6 meters, 7.5 to 8.5 feet. With its claws extended, it would have measured over 3.5 meters, more than 11 feet, making it the largest arthropod ever known. The fossil that revealed Jacalopterus's enormous size was a Chelicera, a claw, discovered in 2007 in the Klurf Formation near Wilverath, Germany. This single claw measured 36.4 centimeters long, but it was missing a portion of its length, suggesting the complete claw would have been 45.5 centimeters, nearly 18 inches. By comparing the proportions of this claw to those of other, more complete Eurypterids, researchers estimated the body size of the animal it belonged to. The result was astonishing. Jacalopterus was not just large, it was gigantic, exceeding the size of all other known arthropods by a significant margin. Jacalopterus belonged to the Pterygatidae, a family of highly specialized predatory eurypterids. These were active hunters with enlarged, robust chelicerae adapted for grasping and crushing prey. The chelicerae of Jacalopterus were particularly impressive, with a curved, powerful structure lined with teeth of varying lengths and sizes. These teeth were designed for puncturing and tearing, allowing Jacalopterus to grip struggling prey and inflict severe damage. Fossilized bite marks on the remains of early fish from the same period suggest that Jacalopterus preyed on armored fish, puncturing their bony plates with repeated strikes. The eyes of Jacalopterus were large and complex, with thousands of individual lenses arranged in compound structures similar to those of horseshoe crabs. Recent studies of fossilized eurypterid eyes have revealed that they had sophisticated visual systems, including structures that enhanced contrast and edge detection, crucial for hunting in murky water. With such advanced vision, Jacalopterus would have been an extremely efficient predator, capable of tracking fast-moving prey even in low-light conditions. Unlike the terrestrial giants, Jacalopterus lived entirely in water, likely in freshwater rivers and estuaries. The deposits where its fossils have been found suggest shallow, brackish to freshwater environments, not the open ocean. This makes ecological sense. Freshwater habitats during the early Devonian were rich with life, including a diversity of early fish and smaller arthropods, providing abundant prey for a predator of Jacalopterus's size. The shallow nature of these habitats also meant that Jacalopterus did not need to contend with the extreme pressures of the deep ocean, which could complicate the physiology of such a large arthropod. The reason Jacalopterus could grow so large in water, while terrestrial arthropods were limited by oxygen diffusion, relates to buoyancy. In water, the body is supported by the surrounding medium, reducing the structural demands on the exoskeleton and allowing for larger sizes without the risk of collapse under gravity. However, Jacalopterus still needed to obtain oxygen, and eurypterids are thought to have had a dual respiratory system, with both gills for aquatic respiration and possibly book lungs or similar structures for air breathing during short excursions onto land. The extinction of Jacalopterus and other giant eurypterids came gradually as environmental conditions shifted. The late Devonian was a time of significant extinction events, affecting marine life more severely than terrestrial organisms. Eurypterids declined dramatically during this period, and by the end of the Permian, they were gone entirely, victims of the same mass extinction that would devastate so many other lineages. The world of the Carboniferous and Permian was a world defined not just by its giants, but by the ecosystems in which they lived, the intricate webs of life that connected predator and prey, plant and decomposer in a dance of energy and survival. The forests of the Carboniferous were unlike any that exist today, so alien in their composition and structure that a modern botanist transported back to that time would struggle to identify many of the plants, would find themselves in a world where familiar plant families were either absent or represented only by primitive ancestors, and where the dominant vegetation belonged to groups that have since declined dramatically or disappeared entirely. These were not forests of flowering plants, which would not evolve for another 150 million years. They were not forests of the conifers that would come to dominate during the Mesozoic and beyond, though early relatives of conifers were beginning to appear. These were forests of giant club mosses, towering lycopsids with names that evoke their strange nature: lepidodendron and sigillaria, trees that could reach heights of 40 meters, over 130 feet, with trunks 2 meters wide at the base, dimensions that rival some of the largest trees alive today. But these were not trees in the modern sense. They were overgrown versions of the small, humble club mosses that still exist, growing on forest floors and in bogs. Plants that in the modern world rarely exceed a few centimeters in height. In the Carboniferous, with the aid of high oxygen levels and the absence of competition from more advanced plants, these lycopsids became giants. Their bark was distinctive, covered in diamond-shaped leaf scars arranged in spiraling patterns, giving the trunks a scaled appearance that earned them the nickname scale trees. These scars marked the points where leaves had been attached, leaves that would fall as the tree grew, leaving behind these permanent marks. The leaves themselves were long and strap-like, tough and leathery, clustered at the tops of the trunks in dense crowns that resembled giant brushes, more than the spreading canopies of modern trees. These trees reproduced by spores, not seeds, releasing clouds of microscopic spores into the air, hoping that a tiny fraction would land in suitable habitat and germinate. It was an inefficient strategy compared to seed production, but in the vast, unbroken forests of the Carboniferous, with favourable conditions everywhere, it worked well enough. The root systems of these lycopsids were as unusual as their above-ground structures. They were shallow and adapted to waterlogged soils, with hollow, spongy structures that allowed oxygen to reach the roots even in the anoxic mud of the swamps, environments where most plants would suffocate. These roots spread wide rather than deep, anchoring the trees in place, but making them vulnerable to strong winds. When storms came through, these giant lycopsids would topple, crashing down into the swamp water below, where they would begin the slow process of fossilization that would eventually turn them into the coal seams that powered the Industrial Revolution. Alongside the lycopsids grew giant horsetails, relatives of the tiny, delicate horsetails that survive today in damp places around the world. Plants that most people walk past without noticing. But in the Carboniferous, plants like calamites reached heights of 20 meters, over 65 feet. Their stems jointed and segmented, much like bamboo, growing in dense thickets, along riverbanks, and in flooded areas where their roots could access constant moisture. The stems were hollow, lightweight but rigid, and they grew quickly, colonizing disturbed areas with impressive speed. Tree ferns added another layer of greenery to these forests, their fronds spreading out in broad, lacy canopies, creating a mid-story beneath the lycopsid crowns. While seed ferns, plants that looked superficially like ferns, but reproduced with seeds rather than spores, formed an understory, their smaller stature allowing them to thrive in the dappled light that filtered down through the canopy. And towering above all, at least in some areas, were the chordates, early relatives of conifers, with long, leathery leaves and a more familiar tree-like structure, their wood dense and resinous, their seeds enclosed in cone-like structures. The ground beneath these forests was a world unto itself, a thick mat of decaying vegetation, constantly moist, often flooded, and teeming with life in forms that would seem both familiar and strange to modern eyes. Millipedes, some small, some reaching the extraordinary proportions of Arthropleura, scurried through the leaf litter, feeding on detritus, breaking down dead plant material, and returning nutrients to the soil. Centipedes, predatory relatives of the millipedes, hunted in the same leaf litter, their venomous claws dispatching prey with efficient brutality. Spiders spun webs in the low vegetation, trapping flying insects, while scorpions like Pulmonoscorpius prowled the undergrowth, their stingers at the ready. And everywhere, in every niche, were insects of countless forms, some feeding on plants, some feeding on each other, some feeding on dead organic matter, creating food webs of astonishing complexity. Amphibians, some of them large and crocodile-like, prowled the shallow waters and muddy banks, creatures with names like Eriops and Proterogyrinus, predators that could reach lengths of several meters and were the dominant vertebrates of the time on land and in shallow water. These were not the small, delicate frogs and salamanders of today. These were robust, heavily built animals with powerful jaws lined with sharp teeth, capable of taking down substantial prey. They spent much of their time in water, where their bodies were buoyant and their movements more graceful, but they could venture onto land to hunt, to migrate, to find new pools when old ones dried up. They represented the cutting edge of vertebrate evolution, the first successful invasion of the land by animals with backbones, and they were still learning the rules of their new environment, still adapting, still experimenting with different body plans and lifestyles. In the air, the griffinflies ruled. They patrolled the open spaces above rivers and clearings, their enormous eyes scanning for movement. Their prey included not only smaller flying insects, but also the plant-feeding Paleodictyopterans, another group of giant insects with piercing, sucking mouthparts, adapted for feeding on plant sap. Some Paleodictyopterans had wingspans approaching 50 centimeters, 20 inches, making them substantial prey. The relationship between griffinflies and Paleodictyopterans has been described as an evolutionary arms race, with prey growing larger to escape predation, and predators growing larger to keep pace. This dynamic, driven by the absence of any larger aerial predators, allowed both groups to reach sizes that would be impossible in a more crowded ecological landscape. The food webs of these ancient ecosystems were complex and interconnected. At the base were the plants, capturing energy from the sun and converting it into biomass. Herbivores, including some insects and early reptiles, fed on the plants, while detritivores like Arthropleura processed the dead material, recycling nutrients back into the soil. Predators, from scorpions and spiders to amphibians and griffinflies, occupied higher trophic levels, and at the very top were the largest predators, those that had few, if any, natural enemies. One of the remarkable aspects of these ecosystems is how vulnerable they were to environmental change. The Carboniferous forests depended on high humidity, warm temperatures, and stable water levels. They were adapted to a world of swamps and wetlands, and when those conditions began to shift, the entire structure of the ecosystem collapsed. This is exactly what happened during the Carboniferous rainforest collapse, an event that occurred around 300 million years ago, near the end of the Carboniferous period. A combination of climate change, tectonic activity, and shifts in sea level led to the drying out of vast swamp forests. The humid tropical conditions gave way to more seasonal climates, with distinct wet and dry periods. The lycopsid forests could not adapt. They died off, and with them went the ecosystems that depended on them. For the giant insects, this was the beginning of the end. The collapse of the rainforests reduced the availability of habitat, fragmented populations, and altered food webs. But the death blow would come later, during the Permian, when multiple factors converged to make the world uninhabitable for creatures that had once thrived. The story of why giant insects disappeared is a story of multiple causes, each contributing to a slow but inexorable decline. Oxygen levels, while crucial for the initial rise of gigantism, were not the only factor, and their fall alone cannot fully explain the extinction of these creatures. To understand what happened, we need to look at the broader environmental and ecological changes that reshaped the Earth during the late Permian and early Triassic. Oxygen levels began to decline during the middle Permian, dropping from their Carboniferous peak of 35% down to levels closer to 25%, and eventually to around 15% by the late Permian. This decline was driven by several factors. The Carboniferous rainforest collapse reduced the amount of photosynthesizing plant life, decreasing oxygen production. At the same time, the evolution of more efficient decomposer organisms, fungi and bacteria, capable of breaking down lignin and other complex plant materials, meant that dead plant matter was now being oxidized, releasing carbon dioxide and consuming oxygen. The balance that had created hyperoxic conditions was broken, and the atmosphere began to shift back toward lower oxygen concentrations. For large insects, this was problematic. As oxygen levels fell, the efficiency of their tracheal respiratory systems declined. The concentration gradient that had allowed oxygen to diffuse deep into their bodies weakened, and larger insects would have struggled to maintain adequate oxygen delivery to their tissues. This would have been especially challenging for active, flying insects like the griffinflies, which required high metabolic rates to power their flight muscles. Over time, selection would have favored smaller body sizes, as smaller insects could function more efficiently in the changing atmosphere. But oxygen was not the only pressure. Another critical factor was the rise of aerial, vertebrate predators. For most of the Carboniferous and early Permian, insects were the only flying animals. The skies were theirs, and size was an unmitigated advantage, with no threat from above. But that began to change with the evolution of gliding reptiles in the late Permian, and more dramatically with the appearance of true powered flight in vertebrates during the Mesozoic. By the Triassic, the first pterosaurs had taken to the air, and later, birds would evolve from theropod dinosaurs. These flying vertebrates were fast, agile, and highly effective predators. They could outmaneuver the large, less agile griffinflies, and they imposed a new selective pressure. Suddenly, being large was a liability. Large insects were easier to spot, easier to catch, and provided a more substantial meal. Smaller insects, with their greater maneuverability and ability to hide in dense vegetation, had a survival advantage. A study published in 2012 analyzed the relationship between insect size and atmospheric oxygen levels over geological time, using a dataset of over 10,000 fossil insect wing lengths. The results were striking. For the first 150 million years of insect evolution, maximum insect size tracked atmospheric oxygen levels closely. As oxygen rose, insects grew larger. As it fell, they shrank. But around 150 million years ago, at the end of the Jurassic and the beginning of the Cretaceous, this relationship broke down. Oxygen levels began to rise again during the Cretaceous, yet insect size continued to decline. This timing corresponds precisely with the diversification of birds and the evolution of more advanced flight capabilities in avian dinosaurs. The implication is clear. Once vertebrate predators occupied the skies, oxygen was no longer the limiting factor for insect size. Predation and competition became the dominant pressures, and insects were pushed into ecological niches where smaller size was favored. The final nail in the coffin for many of the giant insects was the Permian-Triassic extinction event, the most severe mass extinction in Earth's history. Approximately 252 million years ago, a series of catastrophic events, driven primarily by massive volcanic eruptions in Siberia, unleashed devastation on a global scale. These eruptions released enormous quantities of carbon dioxide and sulfur dioxide into the atmosphere, leading to severe global warming, ocean acidification, and widespread anoxia, or oxygen depletion, in both marine and terrestrial environments. The extinction killed an estimated 90% of marine species and 70% of terrestrial vertebrate species. It was also the greatest known mass extinction of insects, with 8 or 9 insect orders becoming extinct entirely, and many others suffering severe declines in diversity. The giant insects, already stressed by declining oxygen levels and increased competition, could not survive this catastrophe. The griffinflies, the giant paleodictyopterans, and many other charismatic groups vanished from the fossil record. The ecosystems they had dominated for tens of millions of years collapsed, and when life began to recover in the Triassic, the world was fundamentally different. The insects that survived and diversified were smaller, more adaptable, and better suited to the new ecological landscape. Today, the largest insects are a shadow of their Paleozoic ancestors. The largest living dragonflies, like the helicopter damselfly of Central America, have wingspans of about 19 centimeters, 7.5 inches, less than a third the size of Meganeura. The heaviest insects, like the Goliath beetle, weigh around 115 grams, similar to the estimates for the largest griffinflies. But they are wingless, ground-dwelling creatures, not aerial predators. The constraints imposed by modern atmospheric oxygen levels, combined with the presence of highly effective vertebrate predators and competitors, have set strict limits on insect size. And yet, the legacy of those ancient giants endures. They remind us that the Earth has not always been as it is now, that the conditions we take for granted, the composition of the air, the structure of ecosystems, the identities of dominant organisms, are all contingent and changeable. The giant insects of the Carboniferous and Permian were not anomalies. They were the natural consequence of a particular set of environmental conditions, conditions that allowed arthropods to push the boundaries of their body plans and achieve sizes that seem impossible to us now. Their fossils, preserved in stone, in coal, in the impressions of wings and the trackways of legs, are windows into a vanished world, a world where the skies buzzed with the wings of predators that would dwarf any bird, and where the forests crawled with millipedes longer than cars. These creatures are gone, but they are not forgotten. Every time a paleontologist uncovers a new fossil, every time a researcher refines our understanding of ancient atmospheres and ecosystems, we come a little closer to understanding what it was like to live in that world, to breathe that air, to witness the rise and fall of giants. And in doing so, we gain perspective on our own time, on the changes we are witnessing and contributing to, and on the resilience and fragility of life on this ever-changing planet. If you found this journey through deep time fascinating, if these images of ancient giants have sparked your curiosity or helped you drift toward a more peaceful sleep, consider liking this video and subscribing to the channel. Your support helps us continue exploring the quiet wonders of the natural world, one ancient mystery at a time. And if you have suggestions for future episodes, or thoughts on the creatures we have discussed tonight, please share them in the comments below. Every question, every insight adds to the conversation, and we are grateful to have you here, exploring these stories with us. Until next time, rest well, and remember that even in the stillness of sleep, the past is alive, waiting to be discovered.